So I think we have a great panel that's going to lead to a lot of discussion, so we ought to get right into it. And rather than have the panel members sit up here where you would have to crane your heads, maybe we should have one at a time get up and speak, and then I'll have, then we'll get the whole panel together. So I'm going to start with Patrick Ryan, who's the Senior Director and Head of Epidemiology Analytics at Janssen. And put on your seatbelt for this one. It's kind of an interesting thing. It's going to give you a lot to think about.
Thank you, Rob, and the organizing committee for letting me come and chat with you a little bit. I'm going to switch over. I'm going to do a live demo of evidence, because why not? We're an evidence meeting. Come on. There we go. Great. Thank you. So the title of this talk is Moving from One Study at a Time to All by All Analyses. And what I'd like to do is offer up a contrast to some of the discussions about how we're generating evidence and how good is the evidence we're generating, and to talk about something that Marcus brought up at the end of yesterday, which is keeping our eye on the ultimate goal of what we're actually trying to accomplish here. So by way of introduction, this work is not mine. This is in collaboration through the Observational Health Data Sciences Informatics Collaborative. Specifically, the work that I'm going to be sharing is largely been led by Martijn Schumi at Janssen, Mark Suchard at UCLA, and George Ribsack and David Madigan at Columbia University. OHDSI is an open science community where anybody can participate in conducting research to try to figure out how can we learn from observational databases. And we have a central coordinating center at Columbia, but this is very much an international community. And our goal is to try to figure out how to improve health by empowering the community to collaboratively generate evidence. Yesterday was largely focused on the challenges here in the US. Within OHDSI, we've deliberately recognized that this is an international challenge. And so within the OHDSI community, we have researchers around the world with very active participation, not just in the US, but also in Europe and the Asia-Pacific regions. Together, there's over 200 researchers in 20 different countries participating across multiple different functions. Much like what you heard from Rich and other groups, we also have a distributed data network where we have open community standards. We have collectively over 60 databases covering 660 million patient records that we're able to conduct observational research on. Part of the reason I'm providing this introduction to OHDSI is it'll probably give you a frame of reference of where I'm coming from the results I want to show. OHDSI's strategy has been that we believe that there's a need to conduct methodologic research, because first we need to establish and evaluate scientific best practices before we apply them. But once we learn those scientific best practices, we think it's essential to do open source analytics development, to codify the best practices into tools that the entire community can make use of. So to Greg's point earlier this morning, it's got to be on GitHub, and everything you're going to see, not only am I going to show you results of stuff that I've generated, but everyone in this audience could generate it too if you had access to data and just downloaded the software I'm going to show you. But most importantly is the idea isn't just about this being an informatics exercise to have data standards and cool software. This is about answering questions that matter to patients. And so the idea is that when we figure out what questions matter to patients, we have I would assert, a moral obligation for us to apply those best practices, use those tools to generate the evidence, and get it out to patients as quickly as possible. Just because I think it might be helpful, we've been framing the different types of analytic use cases that have been important for our community as our focal points for our research. And we've broken them into three categories. Clinical characterization is the basic descriptives of what has happened to a population. We've also done a lot of research in patient-level prediction, so trying to take the experience from others to try to anticipate what will happen to me. And we are also focused on population-level effect estimation, whether you want to think about the safety surveillance or comparative effectiveness, really this question of causal inference. As time allows, I'm going to focus particularly, because the discussion yesterday seemed to be mostly focused on this idea of can we study the effects of medical products. So I'm going to at least start there and dig into, try to describe what we've been observing of where we are today and posit a potential direction forward. So let's talk about population level effect estimation. So I was very interested in the discussion yesterday, and so I figured rather than just talk about this conceptually, I'm going to show you evidence to try to make an argument for where my head's at about this. A comment that Michael Horberg made yesterday really piqued my interest. He said, different people could generate evidence, and on the basis of who generates the evidence changes my opinion about whether I trust it. And in particular, he said, if it's my group in Kaiser, I trust it. If it's some other academic group, I trust it maybe just a little bit less, and if it's industry, I might not trust it as much. So I was just curious about that, and I was thinking the results I'm going to talk about here is depression, just as a particular therapeutic area. And so I just went to PubMed and I just typed in antidepressants Kaiser. And this was the first paper that popped up. It's a paper by Kaiser Northern California looking at antidepressant medication use and the risk of preeclampsia in pregnant women with depression. And in this observational study, they found an observed association associated with antidepressant use, and in particular, they found that the observed association appeared to be stronger for selective serotonin reuptake inhibitors. So is Michael? There's Michael. Okay, so you can trust it because it's your team.
You everything with a certain amount of criticism. I don't say I take everything between rights.
So,
Northern California.
yeah, forget it.
We got these sibling rivalry issues. Goes without saying.
So the basis of this conclusion in the abstract is because they conducted a thoughtful observational study and they observed an increased risk of 1.4 with a statistically significant confidence interval. I'm just circling there. And just to orient you to how I'm thinking about numbers and analysis, I'm just going to represent that data in a slightly different way, and you'll see why in a second. I'm going to draw this picture where on the x-axis I'm showing you the relative risks, so the size, the magnitude of the effect. And on the y-axis I'm going to show you the standard error, so how uncertain we think we are about this association. And what I've drawn here is a dashed red line. That dashed red line represents statistical significance at our magical threshold of p less than 0.05. So if a dot is below the red line, that means it's statistically significant. If it's above the red line, that means it's not statistically significant. So you can see in this case, within this study, there were two analyses. Both were statistically significant, not on the borderline, but they were both statistically significant. So potentially, we might think that antidepressants and SSRIs increase the risk of preeclampsia. A challenge was that there is a different observational study published using the Medicaid data by faculty at Harvard that also looked at this question of antidepressant use and the risk of preeclampsia. And their conclusion was in this population, SNRIs and tricyclics were associated with a higher risk than SSRIs. And very specifically, they point out that SSRI monotherapy has a relative risk of 1, not statistically significant, so a confidence interval from 0.9 to 1.1. So representing this same piece of information on that same picture, what we can see is we've got one study that's marginally significant with a modest increased risk, and we've got another study that shows no associated effect, even though they performed a whole bunch of sensitivity analysis and didn't find anything to be true. Now, I'm of the opinion that we're always thinking about the preponderance of evidence, and it's not so much about whether or not I believe one group's analysis or not, but how do we, in totality, think about this information. And what was interesting is the third paper that I found on this very subject was a meta-analysis and systematic review that actually looked at this question of antidepressants and preeclampsia. And their conclusion, after about 8 years of publications going back and forth, was that although some studies have suggested a moderately increased risk, the current data do not allow for a definitive conclusion on this topic because the studies have many methodologic limitations. In addition, the effect of the untreated depression or anxiety disorder cannot be disentangled from the results. So we're left in this situation that after 8 years, the final conclusion is we don't know. And this is a real challenge that I think we actually need to be able to deal with and address if we're going to think about not this in concept, but how we can actually make use of this information in a very pragmatic way. So, one of the things that, hello, come up. All right. So one of the things that we've been thinking about is, well, what is the magnitude of the evidence that's actually published, not just on depression and preeclampsia, but any disease, any treatment, any outcome? What have we actually learned from real-world evidence thus far? And so what I'm showing you here is a picture where we've mined the published literature to extract out all the observational studies we could find to pull out when the abstract in PubMed provided an effect estimate, a relative risk and a confidence interval, to basically plot what have we seen in the published literature. So we took every paper we could find from PubMed, and specifically here I'm focused only on papers that have to do with depression. And so what you can see here is that same plot. You've got your relative risk on the x-axis, your standard error, and you see all of these dots. Every one of these dots represents a published paper. That yellow dot there, that is our paper from Kaiser, and you can see that it's the same point on that plot. But this pattern itself should be alarming to some of you, because what we're seeing is the totality of the evidence associated with depression. If you just notice the skew of where all of the evidence is in totality, you notice that the dots seem to be skewed a little bit to the right, so they're more positive than they are negative. You see that an awful lot of the dots are below the red dashed line, so they are statistically significant. In fact, 70% of the published studies were statistically significant. You can also see that that dashed line, or a lot of the dots seem to be hovered right at the dashed line. It's almost like someone was kept working at the data until they got p less than 0.05 and then quit. And that's actually something called p-hacking. It's a well-established phenomenon. Now, the issue is that that study from Kaiser might be the right answer for that question. I'm not challenging the veracity of that team or the methods that they've applied. But if that one study is correct, when people are looking into literature to interpret real-world evidence, they're interpreting that one dot in this sea of other dots. And it can't possibly be the case that all of these studies are totally correct if, one, we've got conflicting studies, and two, that the wealth of evidence that's out there actually says that 70% of things are statistically significant, and most of those are increased risks. Otherwise, we wouldn't be doing anything. So that's depression. How does it look like for the rest of the literature? So we actually were able to mine all disease states, and we pulled out 60,000 observational studies that were published. And so that crazy blob of color there, that represents the totality of the evidence from the 60,000 studies that we've pulled out. Still putting that Kaiser dot in that little yellow one so you can see where it fits in this picture. What you see here is that across all disease states, across all treatments, across all causal effects that we were looking for, we observed that 80% of published studies are statistically significant. You still see that positive skew. You see it's all hovering around the dashed red line. And the issue is that many of these studies are probably the best evidence we've got. But when people bring up the concern about whether or not we can trust data, it's not because they're necessarily saying, I don't trust one study. It's that we can't necessarily trust the process that we are using to generate evidence as a community. So we've been wrestling a lot with this question. What do we do, given that currently our process right now is to do one study at a time, where one researcher comes up with one hypothesis, gets access to one piece of data, applies one method to produce one paper, to put one dot on this horrible picture. I got lots of dots up there too. Many of you in the room have dots up here, and I bet you all of our studies are the good ones. But the problem is that we don't know how to tease out the good ones from the bad ones. So what I'd like to offer, one more aside before I offer a potential solution to this. So we've got a bias in the data. Let's also talk about, to the end game that Marcus talked about, let's talk about what we're actually trying to learn for depression. So in addition to all of that evidence from the real world, from the observational data sources, we also have lots of clinical trials. In fact, depression is one of the most studied areas around. And there was a recent meta-analysis of all the comparative clinical trials to basically tell us the totality of what we know about antidepressants thus far. And what's alarming to me is that in their conclusions, they summarized what do we actually know about the comparative effects of antidepressants from clinical trials, not real-world evidence. And there are some things that they feel like we've now finally learned from clinical trials. We believe that sertraline increases the risk of diarrhea. We believe that maybe venlafaxine increases the risk of nausea. But let's look at the serious side effects that at least these authors pulled out. Suicidality, the very side effect that is listed as a black box warning for all antidepressants, their conclusion was that there's evidence from existing studies is insufficient to draw conclusions about the comparative risk. Cardiovascular adverse events, hyponatremia, seizures, hepatotoxicity, all of these, it was the conclusion of the meta-analysis that we have insufficient evidence to tell patients which treatments are going to have a better or worse outcome from all of these effects. So we've got a situation where with the real-world evidence, we're challenged with knowing how we can trust it, and our current apparatus for generating evidence from our randomized clinical trials, we still don't know the answer despite decades of research and hundreds of $millions spent on this question. So, what could we do about that? Well, one of the things that we thought about was, if we boil this down to what would an individual patient want to do? You know, depression is an example where, given the prevalence of the disease, there's a high likelihood that everybody in this room, either you or someone you love closely, is struggling with depression and thinking about treatment choices. I've got someone in my family who wrestles with thinking about, for every time they've got to make a treatment choice, which treatment choices do I want to make, and what considerations should go into that. So if we think about it from the patient perspective, there's 2 or more treatments available for depression, and we want to be able to consider how could we provide evidence for all the things that that patient might consider. So one of the challenges, there's a lot of antidepressants on the market. There's actually, that we focused on, there are 17 different treatments for depression, including pharmacologic treatments and non-pharmacologic treatments. Any patient could be wrestling between the choice of any one of those treatments and any other one of those treatments. So that means we would like to do comparative head-to-head studies for every treatment against every other treatment. Moreover, there's lots of outcomes patients might care about. I might be interested in the question about suicidality and cardiovascular risk. Someone else might be interested in hepatotoxicity and hypernatremia. So rather than me being the researcher and making the choice of the question I want to answer, what if instead we could expose all of the information that every patient would like to have? So we were trying to think about how could we do that. And our idea was that if there was an observational data network of lots of different data sources, standardized, that we could apply best practice methods to, we could answer questions one at a time. So for example here, I could look at multiple databases to ask a very specific question. Does sertraline increase the risk of diarrhea relative to duloxetine? The clinical trials said yes. And what we see here are 4 effect estimates that are all positive, statistically significant. Basically, we're replicating what we've learned in the clinical trial. If I'm interested in the question, does sertraline versus duloxetine increase the risk of suicidal ideation, we can see here that we have 4 effect estimates. None of them are statistically significant. And therefore, I might not necessarily conclude that there is a particular difference for this. Now, this is individual questions. I'm doing one study at a time, and in fact, every one of these little dots could be just like a published paper. You'd be able to drill down to see how many patients were in the study, how many outcomes were observed. We can actually look at the same diagnostics that one would expect to see in a proper published paper, such as using propensity scores to see how balanced the populations are, to look at all of the balance of all the baseline covariates that might be represented. We perform empirical calibration to understand if we were to apply this study design to a series of negative control outcomes, so outcomes that we know are not associated with the outcome, do in fact we observe a null distribution? That is, if we're not expected to see something, can we guarantee that we don't see it and we're not just seeing bias, which is the thing that we've talked about for the last day. We also can perform sensitivity analysis, just like you would do in any published paper, to ask questions about what if you vary the time at risk? Are you still going to see an effect or not? Now, the point here is that if we were to build some sort of a system, you'd like to be able to ask a question for any possible treatment, each possible treatment, or maybe we'd say all treatments, and we'd like to be able to compare that for all comparisons. If we just had available this information, that would be remarkable, because right now this doesn't even exist at all. But how do we know we trust this process that's generating the information? Well, one of the ways that I'll assert that we could learn from this is by thinking about not doing a study one at a time, but by providing the evidence for all treatments against all comparisons for all outcomes. What I'm now showing you here is the results of 17,700 studies that we conducted within the OHDSI study, doing every possible comparison we could do because a patient might be interested in the question. So for every treatment, for every comparator, for every outcome, we have 4 different databases, each one of those following epidemiologic best practices, the same kind of methods that Rich presented yesterday about Sentinel, to apply to this question. But now let's look at the distribution of this picture. It's remarkably different from what we saw from the published literature. Now what you see is that this picture is completely symmetric. Because if A is better than B, B should be worse than A. And if we have an objective source of information, you should be able to see that symmetry. You notice that there isn't anything hugging that dashed red line because we have no p-hacking going on. We've built an objective system that consistently implies best practices to every single question. And oh, by the way, you'll also notice there's an awful lot of dots in the middle that no effect. It's a well-established phenomenon that we know that there's publication bias. It's a lot easier to publish a paper when you find something than it is to publish something when you don't. The problem is, if we don't publish what's not known, we don't know how to trust the things that are known. We don't know what the false positive rate is if we're just not disclosing most of the information. So what you're actually seeing here is that most questions, in fact, 87% of all of these target comparator outcome studies are not statistically significant, provide the full diagnostics for us to be able to explore this. And therefore, we're now left with the idea that for a given patient who has a specific question about one study at a time, they could go get the answer they want by picking whatever treatment they're interested in, comparing it to whatever alternative they're interested in, and asking the question about what outcomes matter to them. However, as a system, we've generated evidence that collectively gives us a higher level of confidence than the current apparatus that we're generating evidence. So I've got 1 minute left. So sorry about that. So I want to make an assertion here that whether you are doing characterization questions, and the simple question is, what is the incidence of a side effect for a drug? Just how often does it happen? That's information that's super hard to get right now. And this large-scale thinking that we could just do it for every treatment and every outcome to provide that evidence, that would be a tremendous resource to patients. Population level estimation, what I just showed you, the idea that we could actually think about causal effects. I would argue that a lot of people are concerned about the idea of we're blindly applying statistics and we're generating lots of numbers and how are you going to trust it? And I'll actually argue the opposite, that we can trust this information a lot more if we take a systematic standard approach than we can by having ad hoc studies one at a time. And then the third area, patient-level prediction, we can apply this same idea because as a patient myself and caring for my loved ones, the questions that usually come up isn't, does this drug on an average population have an increased relative risk of some side effect? The question that I get from my mom and my family is it going to happen to me? And that's actually a prediction question that this same large-scale thinking, we should be able to predict for every treatment what is the risk of a given outcome. And I think if we can apply these standardized approaches to real-world data, not only are we going to generate a lot more data, but it's going to be a lot more credible and a lot more actionable to patients. So with that, I will stop. Thank you.